Good morning, everyone, and welcome to today's webinar, Resolve Complexities in Cancer Research with Single Cell Insight. Thank you all for joining us today, and I'm Meng Thi, the moderator for today's webinar. Before we begin the webinar, a quick note on housekeeping. This session will run for about 30 to 40 minutes, and we will have a Q&A session at the end of the webinar. If you have any questions during the webinar, please feel free to type in the chat box and we will address them after the presentations. For the best experience, please make sure that your microphone is muted throughout the session. This will help to maintain a clear audio for everyone attending. In today's webinar, we are excited to have Mr. Chee Hong as our speaker. He is the Field Application Manager at NextGene Scientific with over a decade of experience in his field. He will be sharing a valuable insights on how single-cell analysis can provide a deeper understanding of cancer research. Without further ado, let's welcome our speaker today. Chee Hong, the floor is yours now.
Okay, thank you, Meng Thi. And first of all, I'd like to thank everyone for joining the webinar today. So Meng Thi, can you hear me?
Yes, we can hear you.
Okay, so now I'll be sharing my slide. So one moment. Okay. Meng Thi, can you see my slide?
Yes, we can see your slides. And everything is good.
Okay, then without further ado, okay, so my name is Chee Hong. I'm from NextGene. Today, our sharing will be on resolve complexity in cancer research with single cell insights. Thank you for joining. So here will be the agenda of today's sharing. So first off, we'll be looking at about what is actually single cell sequencing, and then our service provider, our brand, how does we 10x Genomics make single cell sequencing work? Okay, then after understanding all this theory behind, then we can then proceed to how can we use single cell sequencing and then apply it for cancer biology studies. So in terms of immunogeneity, tumor microenvironments, rare cell discovery, and last off will be continue with looking at the multiomic cytometry using the Feature Barcode technology. Okay, so let's move to the first agenda of today. So which is what is single cell sequencing? So now we shall back to the basic. So which is actually the central dogma. Okay, so everyone know that in central dogma we have the genetic information stored in the form of DNA for most of the organism on Earth. So this particular information will then be accessed. Okay, so normally it's just act as a storage. When we need to use this information, we want to access it, we will then transfer it into RNA. With RNA, we can then produce the functional unit, which is actually the protein. So why I going through this central dogma again is that for using human as an example, so all our body has maybe billions of cells, and all these billions of cells actually has the same genetic material. The coding information is actually the same. The material, the genetic information is the same. However, when we look on those cells, different part of our body, we realize that they are actually different. They are not the same. Okay, so how does it happen is because from the coding information, not all of them will be expressed. So if certain part of our body, certain type of tissue required to use certain function of the stored information in the DNA, it will then only be expressed and produce the functional unit protein. So therefore, different cell, due to their function different, will have a different set of gene being expressed. Okay. So if we want to give it a term for this different set of genes that are being expressed in different cell, we can call it the gene expression profile for the cell. So it's important to note that the RNA profiling to know that what set of gene within a cell, okay, is being expressed is actually the key to have the cellular phenotyping. So traditionally, there are various methods that we can use to study this gene expression. Okay, so the commonly used tool will be the qPCR quantification method. Okay, this method is very good, very well established and reliable. However, it has some limitation. For example, it can only study a couple of genes, so possibly maybe 2 up to maybe 7 or 8, and then could be too much to be handled, okay, for the qPCR. I mean, in terms of the person itself, not so much of the machine. Okay, and then and qPCR is considered targeted, means that you need to select your gene of interest, and then you use qPCR to study only those selected genes. If you have something that is unknown, if you want to look at a comparison between two conditions, and you have so many genes, new gene, novel gene that you want to study, then we have like the older technology, but slightly more, offer more information, that will be the microarray. Okay, so of course, then microarray has been replaced by the NGS technology, the next generation sequencing. Okay. By using next generation sequencing, we can actually do gene expression sequencing and study as well. However, these three method has one thing in common, which is that they does not consider the expression profile between or among cells. Okay. What I'm trying to say is, when we extract RNA for this kind of study, most of the time we are extracting RNA from possibly 100,000 or even million of cells. Okay. So the question will be, are there any variation within this cell? Are we using a heterogeneous cell population? If yes, then they should have a different gene expression profile. And when we actually use pull them together, this information does not show the differences among the cell. Okay. So next off with what is single cell sequencing. So this is the example of what I was trying to share with you guys earlier. Let's say, using an example of blood cell of human, we have neutrophil, basophil, we have eosinophil, T cell, B cell, and all that. Okay. So different cell type has different function, and therefore they have distinct expression profile. However, by using the traditional method, we will actually extract RNA from all these type of cell. Then all these extraction profile, instead of looking in them individually, okay, based on their cell type, we actually average it out and only get one single information. We are like treating them the same, and we are treating them like they should have the same expression profile, but in reality, there are differences. This is an analogy commonly used by 10x Genomics. So the traditional NGS sequencing, we call it the bulk RNA sequencing, is like a blend fruit juice. Okay. So there are so many fruit that we actually blend together and then mixing. So the flavor, just based on it, we can still get the hint of what kind of fruit is being used. However, we are not confirmed. So by using single cell sequencing, we will get the better resolution. Okay. So it's like we are eating just the apple or the strawberry or banana by itself. Then we can have a better separation. So single cell technology actually is not something new. It has been around for more than 10 to 20 years ago. It's just that previously it was a bit laborious, it's not easy to perform. Okay. So before the commercialize of single cell product existed, early single cell publication typically profile less than 100 cells. So techniques for generating single cell data were typically homebrew and challenging. Often you need to use qRT-PCR analysis as well, and only allowing for a profiling of a handful of genes. So commercial solution for single cell RNA sequencing make it easier than ever to generate impact insights. So now we can actually have single cell sequencing of more than 5,000, 10,000, or even 100,000 of cells in the same sample. This is here. I would like to share with you guys the example of a single cell sequencing result. So over here, it will be display cell based on their gene expression profile. So let's say each individual dots over here are actually a single cell. Okay. So by using a clustering function means that set of genes that are similar being expressed between cells, we will actually cluster them together. So this means that all cells here has very similar gene expression profile and but they are actually distinct from, let's say, this set of cells. So this set of cells shares similar expression profile but different from the other set. Okay, so this is actually just an illustration. For the real result, it will become on the right hand side over here. So just imagine that here will be the cluster of cell share same expression profile. This will be another cluster, another cluster. Each cluster maybe have like maybe few thousand cell or 100 cell depending on how many cells that we actually sequence. So the cluster cell actually has similar gene expression profile. For example, T cell, B cell, the one shown over here will be the B cell cluster, the NK cell cluster, the plasma cell, epithelial cell. Yeah. Okay. So next up, how actually the single cell sequencing work. Okay. So the first generation one, we actually using the flow cytometry method. We actually sorting the cell into individual single cell. So one cell go into one well. So let's say over here, the example, we have a 96 well plate. We will have 96 single cell. So if you want to have more, then you need multiple plate or you might need to use 384 plates. Okay. So therefore, by using this method, it's actually low throughput. So with some improvement of technology, we are using the nano well. Okay. So we have a column with nano well. Okay. So in addition to separate cell into single cell, another important thing that we need to bear in mind will be we need to label the single cell with a unique barcode. So what this means is actually that the barcode will be added to all transcript of one single cell. So the second single cell will have different barcode labeled to all of its transcript. So if you have like 1,000 cells, you should have like 1,000 barcodes. So at the end of the day, each of them will be labeled with distinct barcode. Okay. Why we need barcoding them is because during sequencing, we actually need to pull them together. Okay. So after sequencing, based on the barcode, we trace it back to which cell we're talking about. And then we do the clustering, so grouping the similar expression profile cell into the same cluster. So then the nanowell is moderate throughput, but still challenging to scale up. So therefore, we have the combine of the plate and nanowell. And finally, in 10x Genomics, we are offering a microfluidic droplet-based single cell technology. Okay. So over here, we are showing that this is actually engulfed by a phospholipid bilayer. So inside, we have a functional gel bead. The function of this gel bead is actually to have a unique barcode. So this phospholipid bilayer is supposed to only engulf one single cell, and all the RNA in this single cell will acquire the unique barcode from the gel bead. So this particular microfluidic technology make the entire workflow scalable, streamlined, and complete in one day. So 10x Genomics, we offer the end-to-end solution entire workflow for single cell. So we have the gel bead shown earlier, the reagent to label the barcode, and then to create single cell with a unique barcode. Okay. Then once this thing has been ready, we will sequence it, and we offer a free software for the analysis, basically to display a cell cluster with unique expression profile. So here is showing that the sample is going into the microfluidic, okay, and then one gel bead with one sample, it will go through the oil so that it will be engulfed by the oil. So inside one engulfment, there will be only one gel bead with unique barcode, and single cell inside it will be labeled with that unique barcode. Okay, so the application of this will be nearly in all area of biology. So you can do some cell aliasing, co-localization of receptor and ligands pair. It can be used for developmental biology, so identify genes and cells related to morphogenesis, so immunology, neuroscience, and of course the topic of today, oncology. So we can use single cell technology to study the tumor microenvironment, tumor heterogeneity, disease progression, cancer morphology, and of course tumor infiltrating lymphocytes. So how can single cell data apply and help in cancer research? So we can characterize and identify heterogeneous cell population, let's say different mutation of the cancer cell, the tumor, or let's say the normal cell and the interaction of the immune cell to the tumor. Okay, then because we have the gene expression data, we can discover new cell markers and regulatory pathway. We can uncover novel cell type and rare cell type because of the clustering. So we can reconstruct developmental hierarchies and review lineage relationship and profiling healthy and diseased tissue and organs. Okay, so the next topic, the second last, how can single cell sequencing be applied to cancer biology? So single cell technology help us to gain multidimensional view into cancer biology. So we resolve cancer with single cell and multiomics. Okay. So the first thing that we can use single cell for study will be the tumor heterogeneity. We can uncover hidden tumor clones and dissect cell type differences. So we can measure genetic and epigenetic changes on cell by cell basis. So next up, we can see how the tumor cell interact with the normal cell and also the blood cell and all that, and even the immune cell. So therefore, we can study the tumor microenvironment. So it allows us to discover the spatial context of the tumor microenvironment, capture dynamic transcriptional states in a single cell. Next up, we can see how immune cells interact with them. So we have the immune oncology. So advanced immunotherapy by characterizing the tumor immune response, define the receptor repertoire of the infiltrating immune cell. Okay, so how can it apply to cancer biology? So first off, we look at a publication on using single cell to study the heterogeneity of the tumors. Okay. So in this publication by Stewart, is working on SCLC, which is actually the small cell lung cancer. Okay. So SCLC is known to have a robust response to treatment, but when it's actually followed with a relapse, okay, then it realized that it suddenly acquired some resistance. Okay. So therefore, the author using single cell RNA sequencing to look at the tumor heterogeneity before and after the treatment. Okay. He tried to see that what are the differences before the treatment and after the treatment. Okay. So over here on top, these are the example of 3 sample of the tumor. So based on the single cell sequencing result and analysis, we can normally cluster it into 2 cluster of cell. So this one also 2, and this one we have 1, 2, 3 cluster of cell. So therefore, the heterogeneity of the cancer cell are not that many. However, following a treatment and relapse, okay, so when the cancer actually acquired the resistance, so these are the single cell sequencing result of those samples. So as you can see, suddenly the cluster become more, means that the variation between the cell type in the tumor become more. Okay. So here we have 4 cluster, and this one sometimes even up to like 8 to 9 cluster. So when we have different like 9 different set of cluster of cancer cell, each of them might have a unique resistancy, and therefore the drug treatment might not be able to targeting all of them. Okay, so therefore the study from Stewart continue to look at the gene expression profile of all these cluster. So this is before treatment, only 1 or 2 cluster, and after treatment, up to 8 and 9 cluster of them. So they do this particular result did suggest that this different set of expression profile of different cluster actually could have offering the resistance to the drugs, and therefore their suggestion will be that in the preliminary treatment, so multiple drugs combination treatment should be considered to prevent the rise of the resistance of different clusters. Okay. So next up will be a publication to show that how single cell sequencing giving or helping in the research of tumor microenvironments. Okay. So this is a publication by Seth Patty. Okay. So this is a BCC cancer. Okay. And we have the tumor microenvironments. So therefore, in addition to tumor cell cluster, we are able to find fibroblasts, endothelial, myeloid, B cell, NK cell, and other immune cell. Okay. So not only we are able to cluster them, but we can have the information about the gene expression profile of the cell. So by having this gene expression profile of, let's say, T cell and tumor cell, we can actually do a comparison versus a T cell, let's say, from a peripheral blood, to see what are the response of the T cell when they actually are meeting the cancer cell, and how they actually deal with the tumor cell. Okay. So this is another publication by Liu. Okay. So working on the nasopharyngeal cancer. So in this particular study, they profiled 176,000 cells. So 176,000 dots of them. So the total cell number from 10 nasopharyngeal cancer, and it's actually a tumor blood pair. So comparing the tumor and the blood cell. So the transcriptome will be sequenced using the single cell, and here we have the cluster of CD8 cell, T regulatory cell, T helper, NK cell, and so on. Okay. So what this particular study found is that the CD4 and CD8 T cell cluster in the tumor sample had widespread overexpression of exhaustion markers compared to the peripheral blood. Okay, so they are exhausted, and therefore their cytotoxicity is actually become reduced. So the next publication will be giving insight in immuno-oncology. So this study is about the peripheral T cell expansion predicts tumor infiltration and clinical response. So the author assessed various population of T cell and the receptor of this T cell in the tumor, and then compare it to the adjacent normal tissue, and as well as the peripheral blood using the our 10x genomic profiling. Okay. So what they discover is that they are able to separate the clones for the CD8 cell and also CD4. And then they also discover that the dual expanded chronotype are found in the tumor and this normal adjacent tissue were also typically detected in the peripheral blood. So this suggesting that a relationship between the peripheral clonal expansion and infiltration into the tumor and normal cell. So patient with the gene signature of chronotypic dual expansion response respond best to the anti-PD-L1 therapy, and alternative model to the current understanding that immune checkpoint therapy reverse exhausting phenotype of T cell. So, and next will be another publication to share on how single cell technology is being used for the detection of rare and new cell type. So 10x Genomics is able to enhance the detection of challenging cell type, for example, the neutrophils. Okay. And this is an example using the technology to detect a CD8 plus NK cell, which is actually the unique cell cluster in the nasopharyngeal cancer tumor microenvironments. So in this particular study, the author are able to found that the CD8 plus NK cell are present as a distinct cell population in the tumor microenvironment and not found in the normal nasopharyngeal tissue. So this cell have not been previously reported in other NPC study. So they are only observed in other diseases associated with HIV and EBV infections. Okay, so this is another study by Wang and study of the adult human glioblastomas. Okay, so they discovered that it harbor a radial glia-like cell. Okay? So author hypothesized that the RG, radial glia-like cell, could represent the cell of origin or the cancer stem cell residing at the top of the tumor hierarchy and may be key for initiating tumor growth. So another study that after performing clustering and analysis, they've discovered a marker called PAM50 genes, and it could be the signature identified cancerous cell cluster. So the last sharing topic of today will be looking at the multiomic cytometry with feature barcode technologies. So in single cell technology, a lot of times we are looking at just the gene expression profile. In multiomic, we are able to compare the gene expression data to, let's say, the protein labeling, the protein markers by using antibody binding, or even the study of the epigenetic condition of the DNA. So here is actually showing an example of the feature barcode. In the same sample, we can study the gene expression profile using the gel bait. And before we are studying this, we actually label the cell with antibody. So the antibody will bind to the unique marker or cell surface marker on the cell. And this antibody is not a normal antibody, but it actually conjugated with a feature barcode. So during sequencing, we are not only sequencing the gene expression, the transcript, but we are sequencing the presence of the antibody as well. If we are able to recover this unique feature barcode, it means that the cell has been labeled with antibody A, and let's say the other feature barcode representing another antibody B associated with the cell surface protein. Okay. So in addition to protein information in the same set of data, we can actually look at the epigenetic condition of the cell as well. Again, we have the transpose to label the region of open chromatin. Okay, so the TN5 transpose is preferably adding the adapters to the open chromatin, but not the closed chromatin. Okay. So by adding the adapter to the open chromatin and sequencing of the adapter sequence, the feature barcode, we will then associate the region of the DNA to the gene expression profile. So the sequencing of the open chromatin is, we name it as, not we, but the community name is as the ATAC. Okay, so this is showing the epigenomic condition of the sequencing, and then we can compare and associate with the gene expression profile. So by using this feature barcode technology, we can link gene expression and epigenetic data from the same nucleus. So here is actually combining three of the information. So the epigenetic by using the ATAC, the sequencing of the gene expression profile, as well as the protein labeling of the cell surface protein, and we sequence the feature barcode. Then we can compare the cell surface marker, gene expression profile, as well as the epigenetic information in a single set of the sequencing data. Okay, so this is actually showing the real result from Granja and comparing the gene expression clustering versus the epigenetic information in ATAC. So by combining this data type, distal regulatory element could be linked to genes to identify leukemic genes regulatory and expression programs. So the linking of transcriptome and epigenome modalities enable deeper understanding of B-cell lymphoma regulatory network. Okay. So the pair ATAC and gene expression data from single cell and no computational inference required. Okay. So this is a study showing the identification of a tumor-specific gene regulatory network in human B-cell lymphoma by associating the gene expression profile and also the ATAC chromatin accessibility. So reconstruct a gene regulatory networks using both TX accessibility and TF expression measurement in the same cell, and joint measurement of single cell sequencing expression and single cell ATAC sequencing increase confidence in regulatory network. So the Sepati publication again. So identify changes in immune cell abundance after cancer immunotherapy. Okay, so they will actually comparing the responsive patient versus the non-responsive patient to identify exhausted CD8 T cell and helper T cell. Okay, so with that, I will end my sharing of today. So I hope that everyone gets some useful information